Hello, my name is AdamWynn. I'm a second-year medical student here at Texas Tech University Health Sciences Center, and I'm going to be talking about a study I was able to help with over the summer entitled Effects of Consumption of Coconut Oil or Coconut on Glycemic Control and Insulin Sensitivity. It was a systematic review and meta-analysis of interventional trials. So there are a lot of claims in the dietary world about beneficial effects of coconut oil. Some of these include that it's beneficial for cardiovascular health and also for glycemic control. There was a recent meta-analysis that looked at the cardiovascular health benefits and invalidated the claims. But there has not been a meta-analysis looking at the effects of coconut oil on glycemic control. This is still somewhat undecided. So for our study, we decided to do this. We performed a meta-analysis and systematic review that looked at dietary coconut oil's effects on glycemic control. And we looked at both acute and long-term markers, acute being less than 10 days of intervention and long-term being greater than 10 days of intervention. And our hypothesis was that coconut oil would improve the postprandial and long-term glycemic control markers compared to matched controls. And postprandial just means after eating. To perform our study, we first searched the 4 databases listed on the left for relevant studies using specific predefined keywords. That gave us about 2,500 records. We screened for duplicates, and that got us to about 1,300 records. And with these 1,300 records, we screened the titles and abstracts only to see if they met our exclusion criteria. This includes things like non-human studies, full text unavailable, and so forth. And any article that met the exclusion criteria was obviously excluded from the study. This gave us about 51 articles, and with these 51 articles, we looked at the full text to see if there were other things that we had missed or additional things that would make them essentially unable to be used for our meta-analysis and systematic review. And those are listed on the right. Other things include it being a glycemic index study or no control condition, things like that. So in the end, we had 15 studies that we were able to use for our systematic review and meta-analysis. Before we get into our actual data, I wanted to briefly explain how area under the curve works. So if you imagine time 0 here being where you eat a meal, you see how the purple line spikes and then slowly decreases over time. And this purple line could be different things. It could be glucose, it could be insulin, but you'd see they spike after eating a meal and then they go down over time. And we can measure the area under this line. It's known as the area under the curve, and we'll use this in our analysis in the next few slides. So now getting into our actual data here, we've got the studies listed here on the left with their data, and then at the bottom we've got both fixed effects and random effects meta-analysis data. So the main difference between the two is fixed effects tends to put less weight on studies that have smaller sample sizes. But you see highlighted in yellow, in the random effects model, our area under the curve of blood glucose was significantly higher for coconut-containing meals compared to the control meals without coconut. And then continuing along, looking at the area under the curve of insulin, we can see that again, for the random effects model, the area under the curve of insulin is significantly decreased for coconut-containing meals compared to meals without coconut. And that mean difference in confidence interval is again highlighted in yellow in the bottom right corner. We also found that meals containing coconut fat significantly increased fasting blood glucose compared to control meals. And then we looked at fasting insulin, HOMA-IR and HOMA-beta, but those were not significantly different between the two groups. And just so you know, HOMA-IR is a measure of insulin resistance, and then HOMA-beta measures beta cell function, and beta cells are the cells that secrete insulin. So in conclusion, dietary coconut fat seems to be associated with a decreased postprandial insulin response, and this decreased insulin response might be responsible for the increased postprandial glycemic response that we also saw in our meta-analyses. And considering this, contrary to the popular claims, it does not seem that coconut oil has long-term benefits on glycemic control. I'd like to acknowledge the Texas Society of Allied Health Professionals Research Grant 2019, as well as the Medical Student Summer Research Program for helping fund this study. And I've got my references listed here on the right. Thank you for taking the time to listen to my presentation.